everyone, this is the second part of proteins. We will discuss about properties of amino acids and protein structure. Okay, now let's move on the spectroscopic properties of amino acids. In general, all amino acids absorb in infrared region. Only phenylalanine, tyrosine, and tryptophan absorb in UV lights. Absorbance at 280 nanometers is a good diagnostic for amino acids. NMR spectra are characteristic for each residue in proteins, and high resolution of NMR can be used to elucidate three-dimensional structures of proteins. These are UV results from the phenylalanine, tyrosine, in tryptophan in UV region around 280 nanometers. Amino acids joined together become polypeptides. A peptide bond forms by condensation reaction. When the amino acid group or COH and amino group of two different amino acids joins, they release one molecule of water. The hydrolysis reaction is other way around. When a peptide bond are broken by hydrolysis, the hydroxyl group and hydrogen atom from water are added. Proteins are molecules that consist of one or more polypeptide chains. Peptides are linear polymers that range from 8 to 4,000 amino acid residues. The amino acids in polypeptide join together through condensation reaction, and they break down through hydrolysis reaction. This is a peptide bond formation between two amino acids. Since the reaction is a condensation reaction, one molecule of water is removed. The peptide bond is formed between CO carbonyl of one amino acid to NH group to another amino acid. This is an example of structure of a simple peptide consisting of 5 amino acids with 4 peptide bonds. The peptide chain has 2 terminal ends. One is amino terminal end or AN. Another is carboxyl terminal end or CN. The sequence of a peptide is read from N terminal end to C terminal end. Okay, how to name the peptides? For naming peptides, there are several ways. First is using their full amino acid names. Second is using 3-letter code amino acid names. And for longer peptides, as in case in proteins, 1-letter code can be used. This is peptide chain. Since the name is read from N terminal to C terminal, this peptide name is AEGK. Do you know what AEGK stands for? Well, that's your job to find out. Alright, let's move on to the amino acids properties. The amino acids is basically dissolved in water. When dissolving in water, which form occurs? For the basic structure of amino acids, they have non-ionic form and zwitter ionic form. A zwitter ion is a molecule that has both positive and negative regions of charge. In the solid state, amino acids exist as dipolar ions called zwitter ion. Then there is isoelectric point or pi of amino acids. The isoelectric point of an amino acid is the pH at which it bears no net charge. Amino acids have an amino group and a carbocyclic acid. Although they are often drawn in their neutral form in aqueous solution at pH 7 or at physiological pH, their structure is more accurately described as zwitter ion. In practice, the charges on amino acid only balance out to 0 at one specific pH value, and this is called as the isoelectric point or pi. For amino acids with neutral side chains, dpi can be calculated by averaging the pK values of the carbocyclic acid and ammonium groups. For amino acid with acidic side chains, the pi can be calculated by averaging the pK values of the two most acidic groups. For amino acids with basic side chains, The pI can be calculated by averaging the pK values of the least acidic groups. Now we move on to the structures of proteins. There are 4 levels of protein structures. They are primary, secondary, tertiary, and quaternary structure. Any alteration in the structure or sequencing changes the shape and function of the protein. We discuss first about primary structure. The primary structure of a protein is its unique sequence of amino acids. Lysozyme, for example, is an enzyme that attacks bacteria, consists of a polypeptide chain of 129 amino acids. The precise primary structure of a protein is determined by inherited genetic information. At one end is an amino acid with a free amino group, the N-terminus, and at the other end is an amino acid with a free carboxyl group, or the C-terminus. Next is the secondary structure. The secondary structure is special folding of the polypeptide chains in properly arranged repetitive structures. 3 types of secondary structures are alpha helix, beta sheet, and beta turn. Alpha helix structure is formed as a result of hydrogen bonds between the carbonyl oxygen C double bond O and the amide hydrogen NH of the polypeptide chains and does not depend on the side radicals. The shape of alpha helix is like spiral. The next secondary structure is beta sheet. Beta sheet can run in 2 directions, anti-parallel and parallel beta sheets. Both models are found in proteins, but the anti-parallel structure is more stable than the parallel beta sheet. Beta sheet shape is analog to the sheet of piece of paper. The last secondary structure of protein is beta turns. Beta turns permits the change of direction of the peptide chain to get a folded structure. Beta turn loops allow for protein compaction, since the hydrophobic amino acids tend to be in the interior of the protein, while the hydrophilic residues interact with the aqueous environment. These are summary of all secondary structure in proteins. They are alpha helix, beta sheets, and beta turn. The tertiary structure of proteins complete 3-dimensional shape of a given protein conformation. The tertiary structure represents the spatial relationship of the different secondary structures to one another within a polypeptide chain, and how these secondary structures themselves folding into 3-dimensional form of the protein. Tertiary structure is based on various types of interaction between the side chains of the peptide chains. This interaction can be hydrogen bonds, hydrophilic interaction to water, hydrophobic interaction, disulfide bonds, and salt bridge. The last protein structure is quaternary structure in proteins. In proteins, there are some definition. Monomeric proteins are built of a single polypeptide chains, while oligomeric proteins are built of more than one polypeptide chains called as subunits or monomers. The quaternary structure describes the joining of 2 or more polypeptide subunits. The subunits each have their own tertiary structures. The bonds that involving here are non-covalent interaction. Subunits can either function independently or work cooperatively. Dissociation of a subunit results in loss function of protein. Alright, let's recap the 4 protein structures. They are primary, secondary, tertiary, and quaternary. Okay, now you already learned about proteins. See you on the next video on our next biomolecule.